I am Dr. Sonia Sasidharan, Assistant Professor, Department of General Pathology. I will be discussing the topic metastasis. Metastasis is defined as spread of tumor by invasion in such a way that discontinuous secondary tumor masses are formed at the site of lodgement. There are different routes for tumor metastasis, and these include lymphatic spread, hematogenous spread, and spread along body cavities and natural passages. First, about lymphatic spread. Through lymphatic spread, there will be lodgement of malignant cells in the lymph nodes. Lymphatic spread occurs through either lymphatic permeation, that is, lymphatics are readily invaded by cancer cells and may form a continuous growth in the lymphatic vessels, or through lymphatic emboli, in which the malignant cells may detach to form a tumor emboli so as to be carried along the next draining lymph node. Regional nodal metastasis. This means that the regional lymph nodes draining a tumor or the tissue containing the tumor are invariably involved in the metastasis. For example, involvement of axillary lymph node in carcinoma breast. Skip metastasis means sometimes the lymphatic metastasis do not develop first in the nearest lymph node due to venous lymphatic anastomosis, and it may appear in another lymph node. Retrograde metastasis means the tumor cells spread against the flow of the lymph. Examples of lymphatic spread includes carcinoma breast metastasis to axillary lymph node and papillary carcinoma thyroid metastasizing to cervical lymph node. So this is a picture showing lymphatic metastasis. Here we can see a lymph node, and we can see this lymph node is involved or show metastatic deposits from a cancer. Now coming to hematogenous spread, as the name indicates, it is the metastasis which occurs through blood and mainly via veins. Most of the hematogenous metastasis or hematogenous spread of tumors occurs through vein because arteries are more muscular and difficult to get invaded by the tumor cells. The common sites of metastasis which occurs through hematogenous spread are liver, lungs, brain, bone, kidney, and adrenals. Grossly, the blood-borne metastasis in an organ appears as multiple rounded nodules of varying size scattered throughout the organ. This helps us to differentiate the metastatic deposits or secondary deposits from a primary tumor of the organ, because primary tumor usually appears as a single large mass, while metastatic deposits in an organ appears as multiple small nodules. Microscopically, the secondary deposits or the metastatic deposits generally resembles or reproduce the structure of the primary tumor. Examples of hematogenous spread include carcinoma colon metastasis to liver, and follicular carcinoma thyroid metastasis to bone. So this picture shows the gross and histopathological appearances of liver in secondary deposits. Liver showing secondary deposits or liver metastasis. So here we can see in the gross picture that the liver show multiple small nodules of tumor, and in the histopathology also we can see the liver, normal liver parenchyma on one side of the picture and a metastatic deposit on the other side. Now next is spread along body cavities and natural passages. Among this, the main one is transcelomic spread, where the tumor cells invade along the serosal lining of coelomic cavity and spread along the coelomic fluid to get deposited elsewhere in the body cavity. Examples include carcinoma of stomach sealing to both ovaries or the Krukenberg tumor, carcinoma of bronchus and breast spreading to pleural and peritoneal cavities, and pseudomyxoma peritonei. Next is spread along epithelial lined surface, and the example for this is carcinoma of endometrium or ovary spreading through fallopian tube to the endometrium. If the carcinoma is occurring in endometrium, it can spread to the ovary through the fallopian tube, and if it is occurring in the ovary, it can spread to endometrium through the fallopian tube. Next is spread via cerebrospinal fluid, and the fourth one is implantation. That is the spread of tumor through surgeon's scalpel, needles, sutures, and direct prolonged contact. Example for implantation is carcinoma of upper lip spreading to lower lip by constant contact. Now we will discuss the mechanism and biology of invasion and metastasis. Metastasis involve making the passage by the cancer cells to the vessels by dissolution of extracellular matrix at 3 levels. At the basement membrane of the tumor itself, at the level of interstitial connective tissue, and basement membrane of microvasculature. Sequential steps involved in tumor invasion or metastasis include: first is aggressive clonal proliferation and angiogenesis, in which a subpopulation of rapidly proliferating monoclonal tumor cells acquire the characteristics to complete the steps involved in the development of metastasis. And the new vessels formed to support the tumor growth are more vulnerable to invasion by cancer cells than the normal vessels. Second step is tumor cell loosening, in which the cancer cells will loosen from the rest of the cells by inactivation of cell adhesion molecules like E-cadherin. Third step is tumor cell ECM interaction, in which there is attachment of cancer cells to the ECM proteins or extracellular matrix proteins, laminin and fibronectin, and there will be loss of integrins which facilitates invasion. Fourth step is degradation of extracellular matrix. Here the tumor cells overexpresses proteases and metalloproteases, that is matrix degrading enzymes. These enzymes dissolve extracellular matrix at the basement membrane of the tumor cells, interstitial matrix, and basement membrane of the vessels. Fifth step is entry of tumor cells into the capillary lumen. Here there is migration of tumor cells into the vessel lumen, which is facilitated by the autocrine motility factor or cleavage products of the matrix components. Sixth step is thrombus formation. That is, once the tumor cells have entered the lumen of the capillaries, they will get covered by the constituents of the blood to form a thrombus. This thrombus formation helps the tumor cells to get nourishment as well as protects them from immune attack of our body. Next is extravasation of tumor cells. The tumor cells will travel through the circulation up to a point at which it will mechanically block the vessel and attach to the vessel endothelium and then extravasate to the extravascular space. The 8 steps involve survival and growth of metastatic deposit. The extravasated tumor cells on lodgement in right environment grow further under the influence of growth factors produced by the host tissue, tumor cells, and cleavage products of the matrix proteins. Thank you.